So I have the great honor to introduce our speaker, Dr. David Pepin from Harvard Medical School today. Dr. Pepin is a leader in reproductive biology, specifically within the field of ovarian biology and clinical applications. He has a long list of accolades, and I'm not going to be able to list them all, but some of the most interesting recent findings relate to how anti-Mullerian hormone, or AMH, has multiple implications in women's health. His group is one of the first to study how AMH, when in supraphysiological levels, can be an effective contraception. So I hope to hear a little bit about that today, and I'm excited about that. But also, his group studies how AMH could have implications in fertility treatment, understanding reproductive aging, but potentially also serve as a protective agent for people undergoing chemotherapy. And I always like to say, for an amazing scientist, part of it is to have a curious mind and an adventurous spirit. I had something else before, but yesterday we had dinner with Dr. Pepin at a Thai place, and it really speaks to his adventurous spirit that he ordered the durian black rice for his dessert course, and he finished a lot of it. So without further ado, please join me in a warm welcome to Dr. Pepin from Harvard.
Thank you, David, for this warm introduction. I thank the organizers for inviting me. It's an honor. I'm quite happy to be talking to you both virtually and in person here about some of the work my lab has been doing around AMH. And then this is sort of a whirlwind tour of a bunch of different applications and projects we have in the lab. So I'm going to hope to give you little vignettes of where I think AMH may be useful in the clinic eventually one day. I mean, I'm still working on a lot of these details, but I think there's a lot of potential here. So these are my disclosures. Particularly, I'm a co-founder and scientific advisor to Therapeutics, which is developing some of those therapeutic applications I'll be talking about today. So this is basically what my lab does. At the center of it, we're an ovarian development lab. So we do basic research, and a lot of what we're working on surrounds the hormone, anti-Mullerian hormone, and its receptor, AMHR2. And then, on one side, we're trying to figure out what it does, how it contributes to ovarian development, to follicle development. And on the other, whatever we learn about how it works, we try to apply it to clinical problems. And I'll give you a preview of some of the things. So I won't talk today about ovarian cancer, but I will talk a little bit about the three other applications I've listed there. So we're developing therapeutics, different therapeutic modalities surrounding AMH, including gene therapy. And you'll see some data there where we're developing a contraceptive for use in pets, where we're working on recombinant AMH analogs with various applications in terms of oncofertility, and you'll see IVF procedures. And then we're developing small molecule agonists, particularly as contraceptive for women, and potentially even a treatment to reduce the side effects of menopause. And I'll talk about that briefly at the end. So I'll give you an overview of some of those research projects, including many unpublished data. So I'll go back a little bit in time and tell you about anti-Mullerian hormone, or Mullerian inhibiting substance. It gets its name from its function during fetal development, where it's a testicular hormone that inhibits the development of the Mullerian ducts, which of course gives rise to the fallopian tube and uterus and cervix and the upper third of the vagina. And the initial discovery of the function of AMH, not the hormone itself, because that took a little bit longer to identify, but its presumed function was inferred by Alfred Jost, a surgeon in Paris, who was doing these very meticulous dissections where he would transpose ovary and testes between fetuses of rabbits. And then he noticed that if you put a testis, and replace the ovary, that you'd get regression of the Mullerian ducts. And then he put in a testosterone crystal and showed that this could not recapitulate this effect. So there had to be another testicular hormone. And this was done in basically right after the Second World War in 1947. And it took a long time before this hormone was actually discovered and cloned. In 1986, two back-to-back publications, one by Patricia Donahoe, a surgeon at MGH and my postdoctoral mentor, and another woman in Paris, Natalie Josso, also a clinician, and they both coined different terms for the hormone. Pat called it malaria inhibiting substance, and Natalie called it anti-mullerian hormone. And in the end, anti-mullerian hormone sort of stuck as the name for this hormone. So you're all familiar with AMH. AMH is produced by early growing follicles. If I can point here, there you go. So mostly, early secondary, early antral follicle produce the bulk of the AMH in circulation. Very large antral follicles produce very little. And then AMH levels go down as women age, and it's supposedly reflective of the ovarian reserve. But in truth, what it's really measuring is the number of early antral follicles that are producing the hormone. And this is a kind of surrogate marker for ovarian reserve. And then I think part of the reason why the name AMH stuck out is that it sounds a lot like the other gonadotropin hormones, FSH and LH. And in some ways, it's, I think, useful to compare them because FSH and LH are pituitary hormones, unlike AMH, which is an ovarian hormone, but they all together control different stages of the follicle development. So you all know FSH, enhances the growth of early follicles, and LH eventually induces ovulation and luteinization. And AMH acts far upstream in a couple of different ways. One is that it is a feedback inhibitor of primordial follicle activation. And then second, as I'll show, it actually reduces or suppresses the development of early preantral follicles. And we often devote a lot of time thinking about FSH and LH, in terms of regulating follicle development, but they really just regulate the last few weeks of a follicle's life. Most of the follicle's life actually precedes this and is gonadotropin independent. And this is really where AMH has its action. So AMH, unlike gonadotropins, is a very large hormone. It's in the TGF-beta superfamily, and it's a very unique hormone in many respects. So like all TGF-beta family members, it's actually initially produced as a precursor that's inactive. So it needs to be cleaved to be activated. Now, many other TGF-beta family members can form heterodimers, like inhibin, activin, and many of the BMPs. AMH actually only dimerizes with itself. So it forms this complex here. And it's really unlike any other TGF-beta family members. And the second way is that usually the complex of the prodomain, so the larger part of that structure, in the mature domain, the one that actually binds the receptor, usually that is inactive in TGF-beta family members. For AMH, actually, this is the active conformation. So it has its own receptor. AMHR2 is the only TGF-beta family member that works this way. There's one ligand, one receptor. It's a perfect pair, and then it signals downstream. And it has a very unusual prodomain, so it really sticks out. And we have a publication actually that was just accepted last week in PNAS, where we actually described the evolution of that prodomain in collaboration with Tom Thompson at the University of Cincinnati. And it basically is the result of an insertion hundreds of million years ago that made really AMH very unique in terms of structure and function. So another advantage of AMH when we're thinking about hormones that control reproduction is that unlike, say, gonadotropins or especially steroid receptor, AMH receptor is very uniquely expressed in only a few tissues. It's expressed in the adrenal, the ovary, and the testis. So what these three have in common is they produce steroids. So it seems to be a conserved function in regulating cells that produce steroids. And it's really not, the receptor is really not expressed in most other tissues, unlike the estrogen receptors, say, or the progesterone receptors. And then if we look at the ovary itself, the main structures that express a lot of the AMH receptor are follicles, and it goes all the way from primordial follicle to large antral follicle. The expression of the receptor diminishes a little bit in these large follicles, and there's also expression in the surface epithelium and to a certain extent in the stroma and theca. So I'll describe a couple of different technologies on how we try to produce therapeutics aimed on the AMH-AMHR2 axis, and then some of those applications after. So the first problem we had with AMH is that as I mentioned, it's a very large protein. It needs to be cleaved to be activated, so it's very difficult to produce. And then when we tried to produce that using just a recombinant wild type sequence, most of it was inactive. So some of the technologies we developed basically was to optimize the protein structure to allow more cleavage. And so we've called this a new engineered AMH variant, LRMIS, back to its old name of MIS. And then the second technology that we developed was basically gene therapy. So now taking this new analog of AMH, putting in an adeno-associated virus so that we can now infect muscles in our case, because we're using AAV9. Those muscles then produce the AMH protein. It allows us to do very long in vivo experiments where we can have sustained levels of supraphysiological AMH and then see its effect on the ovary. So we don't infect the ovary, as I'll show you, but we do affect the ovary by having high levels of AMH. And then this is what we observe. So after about a month after infecting a mouse with this construct, so a gene therapy driving expression of the AMH receptor, we see the ovary shrinks. It's very much smaller. And if we look at the histology of the ovary, this is a control ovary. You see in the cortex, the primordial follicles that are dormant, and you see many growing follicles here in the middle that are inhibin alpha positive. And then in contrast, when we treat with large amounts of AMH for a month, you can see that most of the follicles are dormant in the cortex. And we have a few that are growing, but very much fewer. And because you have such a small number of growing follicles, the ovary is a lot smaller. So I'll give you the mechanism first, and then I'll show you how we came to these conclusions. So again, what we knew going into these studies is that AMH was a feedback inhibitor, is produced by these early growing follicles and inhibited the activation of the primordial follicle. But then some of the work that we've done showed that also it reduces the rate of maturation of early preantral follicle. And then if you reach very high supraphysiological amounts, what you find is that those follicles stall. So not only are you reducing the entry of the follicle from the quiescent dormant pool into the growing pool, but this growing pool actually stops developing. It's in some ways sort of frozen in time. So how can we use this to achieve contraception? So I'm sure you're all familiar with the current hormonal contraceptives, where they're trying to disrupt the gonadal pituitary axis, the feedback between estradiol and LH. GnRH antagonists are not typically used as contraceptive in humans, but they're often used as contraceptive in animals. Again, you're trying to disrupt gonadotropins. And then AMH is upstream of this. Again, it can act on the early gonadotropin independent stages of the follicle and suppress both activation and growth. It has also activity on the hypothalamic pituitary axis, although that is not the mechanism of contraception. And it also has activity on the large antral follicle, although large antral follicles are much less sensitive to AMH. They actually start downregulating the receptor, and that is also not the mechanism of contraception. So, there's many different graphs that I could have shown here, but they all say basically the same thing. So this is levels in the blood of AMH following infection within the AAV, driving overexpression of the AMH receptor, overexpression of the AMH ligand. So you can see within a week, you're reaching microgram per ml levels. Again, if you're familiar with AMH, normally it's in the low nanogram per ml concentration in the blood. So we're achieving levels that are 100 to 1,000-fold higher than endogenous. And then in this particular case, we had two different concentrations of virus. And then after about a month, I showed you some pictures of histology previously. You can see that the ovaries are much smaller because you have essentially no growing follicles. And you can see that quantified on the right here. So the number of primordial follicles remains more or less the same. In fact, if we wait a little bit longer, you start to have a little bit of an excess primordial follicle because you're preventing their activation. And then if we look at each subsequent stage, there's about a 50 to 60% reduction in primary follicles. Again, you're inhibiting that entry into the growing follicle pool, and you have almost no secondary and antral follicle there. And the ones that are there are sort of paused or stalled. So maybe the most informative experiment that we did was to look at the effect of gene therapy with AMH in a mating study in mice. So the way we did this is we took a group of females and we gave them AV9 AMH. We waited 3 days for the AMH levels to be supraphysiological, again in the microgram per ml range. And then we put these females with males and we looked at their production of pups over time. And you can see that here. So this is the cumulative number of pups. What you'll notice is that actually even treated females will have at least 1 litter. So that tells us a couple of things. It tells us that these females with supraphysiological levels of AMH can ovulate, they can get pregnant, they can bring their pregnancy to term, give birth to those pups. Those pups are healthy, they're normal sex ratios, their learn development is completely fine. But then they'll never have another litter again. And so, that already informs us a little bit about the mechanism of action. So we're not actually inhibiting ovulation. What we're inhibiting is the replenishment of the antral follicle pool that can then ovulate. And that takes approximately 4 weeks, just a little bit over 3 weeks, actually. And then from then on, this animal is completely contracepted and will never have another litter in its life. Now, if we look at steroid production, surprisingly, despite the fact that you don't have cyclic recruitment of antral follicles, you do have normal levels of estradiol being produced for the most part. They're slightly suppressed, but still within the normal range. So these animals behave in terms of sex steroids more or less normally, but they are completely contracepted. And so a couple of years ago, we want to look a little bit deeper into how is it that these follicles are A, inhibited from activating, and B, inhibited from developing once they've activated. And so we use single-cell RNA-seq technology to try to answer these questions. And what I'm showing you here is basically a Tisney plot of representing the gene expression in granulosa cells. And I've overlaid here with 2 markers of granulosa cell activation, so inhibin alpha and NR5A2. And so you can look in the control here, and granulosa cells, when they activate, turn on these 2 genes. And you can tell if you, in this cluster, the ones that are subjected to supraphysiological levels of AMH, very few are able to turn on these 2 genes that are indicative of the activation and growth of the follicle and the differentiation of the granulosa cell. And then you can sort of see it here in these 2 gene signatures. So this is a differentiation signature, including many different genes, including inhibin alpha here. And you can see that the granulosa cells that are treated with AMH, fail to turn on the genes that they should as granulosa cells. So there's an inhibition of their differentiation. And then conversely, if you look at this quiescent signature, many genes that we find in quiescent primordial follicles, even in the control, they're actually even higher in the AMH. So in some ways, what we've done is we've taken this population of primordial follicles and made them hyperquiescent, very dormant. And then the ones that do activate, we're putting the brakes on by preventing them from differentiating normally into granulosa cells and growing. I'll show you a couple examples of that. So, the function of AMH on primordial follicle activation was already known before I did these studies. In fact, if you look at the knockout animal for AMH, it has more growing follicles early in its life. So, we already knew it was a break. So basically, this experiment did the opposite. So now what if we have too much AMH? Now we have a little bit more of a break, and you can tell fewer primary follicles start to grow, and fewer primordial follicles turn into primary follicles. About a 50% reduction in the rate of activation. Again, those that do sort of escape this hyperquiescence are not differentiating normally. And then you can see that here in these ovaries, 1 week after treatment, and these are actually early postnatal ovaries, so they're not in adults, but the mechanism is essentially the same. So you can see in the medulla of the ovary, these larger follicles that are starting to grow, they're turning on their markers like NR5A2, and then we do have a couple of follicles that that activate in the medulla, very much fewer, and they're not growing. You can tell it's only 1 or 2 layers of granulosa cells maximum, and they're not turning on many of these genes. So we're blocking their differentiation. And finally, we're also blocking cell division. So, I showed it in 2 different ways here. Ki67 stain is a marker of cell proliferation. So when you treat with AMH, there's a lot smaller percentage of granulosa cells that are dividing. And this is not the marker top 2A. This is at the single cell level. There's fewer granular cells that are top 2A positive. So we're not only preventing their differentiation, we're also preventing their proliferation. So these follicles are not growing. So a question that we had is now, is this stalling reversible? And we dug a little deeper into the program that was associated with the pausing of these activated follicles. And what we found was an intriguing a signature here of CDK inhibitors. So these are typically inhibitors of cell cycle. And CDKN1A, 1B, and 1C were all upregulated when you treat with AMH. And there's also transcription factors that we know are involved in activation of follicles, or the repression of the activation that are also upregulated with AMH. And so we did this experiment where we treated for 10 days and postnatally. And this is a great time to study primordial follicle dynamics because you have a lot of primordial follicles and they're all kind of activating at the same time. So we treated it with AMH, and it causes those follicles to pause and turn on these cell cycle inhibitors. And you can see an example here of CDKN1B really highly expressed in these follicles. And then you stop treating with AMH and you see, is this signature reversible? You can see within 3 days, the staining is reduced. Within 5 days, it's almost completely gone. And that's true of most of these markers. And conversely, you see these granular cells re-entering the cell cycle. So TOP2A and Ki67, the two markers of proliferation, are turning back on. And then the cell cycle inhibitor, CDKN1A, for example, is coming right back down. So this stalling of follicles appear to be reversible as long as you remove the AMH stimulus. So this is an overview of that mechanism. And so what we found is that the old way to think about it was that primordial follicles are sort of, the activation is dichotomous. You're either quiescent or you're activated. But in fact, if you look at it transcriptionally, what we find is there's a population of primordial follicles. Some are more quiescent than others, and AMH is really pushing them towards that hyperquiescent state. And then once it activates, it's actually also not sort of an inexorable forward motion in development. There's actually a potential to pause the growth of these follicles with AMH, and this pause appears to be reversible. And then these combined actions are what actually prevents ovulation by limiting the renewal of the large antral follicle pool. Now, we have an idea about the mechanism. How can we apply this? So one of the first applications is to veterinary medicine. If I had been told, 15 years ago that, I would be developing contraceptives in cats, I would have never believed it. But this is sort of where the research led us. There was a big need for a non-surgical alternative for contraception. Right now, the way that cat and dog overpopulation is controlled is through spaying and neutering. So trap, neuter, and return. But this is very difficult. You need trained skilled vets to do this, and you need a lot of equipment. And there's over 480 million unowned domestic cats worldwide. So this is a major problem, and it has led to they kill annually 2.4 billion birds just in the United States, and it's led to the extinction of over 33 species. So it is an environmental problem as well, not just a humane problem. And so we've used our knowledge of gene therapy with AMH to apply it to cats. And so we've done a cat version of the transgene. We did multiple different studies. The one I'll show you here, we treated these cats with AV9, and then we did 2 mating studies at the 1 year and 2 year time point. And again, what we're inducing is microgram per ml levels, so about 1,000-fold higher than endogenous, and then putting them with a male and seeing, are they contracepted or not? So how do we follow hormones in cats? We wanted to have very granular data points, be able to follow throughout their lifetime. And we can't take blood samples every day. So we've learned a little bit from conservation biologists and by measuring hormones in feces. And so the way we do it is each cat is given a feed with either dyes or food-safe glitter. So we can identify the feces from each cat. And then every day they're collected and analyzed for fecal steroid metabolites. And this way we were able to get over 20,000 data points on both estrogen and progestogen metabolites. And that allows us to have a really good idea of what's happening in terms of the cycle in these cats. So this is a control cat, just to give you an idea. You have the estradiol in green and the progesterone in purple. And then when you introduce a male, you can tell there's a an estrous phase followed by a luteal phase. So there was a mating here, probably a non-productive mating. Then there's another mating here, an estrous phase followed by a luteal phase, and then that cat got pregnant. And then you remove the male, and there's no more luteal phase. Second mating study, the first try, she gets pregnant again. So this is what happens in control animals. Here is one of the high dose animals. So you can see that there's still estrous phases. When you introduce a male, there's no luteal phase, and this animal did not get pregnant in both mating studies. What's interesting about this particular animal is, cats are usually considered to be induced ovulator, means they need to mate to ovulate, but many cats are actually spontaneous ovulator, and this one was Abigail. You can see that previous to her treatment, she would actually spontaneously ovulate quite often. And after treatment, she never spontaneously ovulated again. Again, further reinforcing the fact that AMH precludes ovulation. And there's different ways to measure that hormonally. So we know luteal phase frequency is decreased with treatment, and progesterone is decreased because the progesterone is associated with the ovulation and the luteal phase during pregnancy. So overall, progesterone is slightly decreased. And then really the proof is in the pudding here. So if we look at litter sizes and number of litters, all AMH treated cats were contracepted in both mating studies. So we achieved complete contraception. And again, interestingly, estradiol levels were not suppressed by the treatment. In fact, they're quite similar to the control group. There's a slight elevation, probably has to do with the introduction of males or maybe seasonal variation. So where are we with this? We're actually are starting clinical trials with the FDA through the CVM, the Veterinary Medicine Department. And this is actually the first gene and therapy product that will be undergoing clinical trial at the FDA. So there's a lot of different things we have to figure out. For example, are these animals safe to go back to their owners, and what's the environmental impact? So it has been a very interesting journey. We're very excited to launch the pivotal studies. Now, what about application to human contraception? So gene therapy is great, but it's not easily reversible, right? Once after injection, more or less have steady supraphysiological levels, and it could be an alternative to tying the tubes or some kind of permanent solution. But really what we want to do is produce a contraceptive that's comparable to current oral contraceptives, but that would have a completely different side effect profile, hopefully a better one, because the expression of the receptor is so limited to the ovary. And so to do that, we need a small molecule, something that would be orally available that you could take just like the regular contraceptive pill. And we've harnessed some of the resources at the Broad Institute, basically using robotic-based drug screens. And these are based on a cell-based assay where essentially AMH signaling triggers luciferase production. So we can look for compounds that turn on AMH signaling and turn on luciferase. And this is an example of AMH doing that here. And we've done several screens. I can show two here, a repurposed drug library of about 5,000 that yielded 16 hits, and a sort of kinase-directed compound library of about 12,000 compounds, where we had 762 hits. So now these are tool compounds. They're not drugs, but they're a good starting point. One of our favorite tool compound there, called it MGH0, we actually tested in vivo in mice. So again, it's nice to use neonatal mice because they're smaller, so you don't need as much. And also, they have all their follicles start growing at the same time, so you can look at its effect on follicle growth much sooner. So it only takes a few days to see whether they're suppressed or not. And then we have transgenic mice that are either deficient in the receptor of AMHR2 or have a working copy of the receptor. That allows us to test whether the effect is specific. So if it's just killing follicles, it'll kill follicles in both. But it's specifically inhibiting follicle development through the receptor, then we should only see it in mice that have a working copy of the receptor. And this is exactly what we see, a suppression of primary follicles with both the AMH protein and the small molecule, only in mice that are wild type, and not in mice that are knockout. suppression of secondary follicles, suppression of tertiary follicles. So this compound is effectively acting just like AMH. But now we needed to make it better. It's a tool compound. We need something that's, you need small amounts that would have as little side effects as possible. And we're developing this in collaboration with Tom Thompson. He's a structural biologist at the University of Cincinnati, with whom we've crystallized and solved the structure of AMH and its receptor. And he's helping us basically tweak these compounds to make sure that they're also very specific to the AMH receptor. We don't want them hitting the other TGF-beta-like receptors, BMPR2 or ACVR2A and B. And so these receptors have subtle differences in their kinase domain. And we want to make sure these compounds are effective only on turning on AMH receptor and not interfering with the other receptors. And so we've done multiple medicinal chemistry campaign. I can show you two lead candidates. Here we have MGH103 and MGH104. And our starting compound is here, MGH0. And then the further you go to the right, the more specific you are. So we're detargeting it towards, here it's aurora kinase. It's one of so that we can reduce side effects. And then and here is the EC50 to AMHR2. So the more you go up, the more potent the compound is on the AMH signaling. And so you want to find compounds that are in the top right corner here. So MGH103 and MGH104 are about a log better at potency and a log better at specificity. They're capable of inducing regression of the Mullerian ducts, so we know they're biologically active. So we're inching slowly towards a drug. I say we're about halfway here. If we can get one more log to the right and one more log up, I think we have a working drug. And then I'll show you a couple. So not only would this be a contraceptive, but just like current oral contraceptives, there's other different applications that you can use this for. And one that you might find of interest is, can we use this for controlled ovarian stimulation? So here's the sort of the idea. AMH, as I showed you, restricts the activation of the primordial follicles, and it stalls the development of those early preantral follicles. So if you have an excess AMH, what ends up happening is eventually you run out of growing follicles. So you have only these early stage follicles that are kind of arrested there. So now what happens when we remove the AMH is not only do you get greater recruitment from the primordial follicle pool, because now you have actually a zero AMH environment. All the AMH came from your treatment, and you have no growing follicles, so there's no more negative feedback. So not only do you get more recruitment, but now also all the follicles that were stalled begin to grow together. And what it gives you is a coordinated wave of follicles that will reach every stage in a very sequential manner. And so the idea is that can you use that to your advantage to time your control stimulation and get greater recruitment. And then, so this is an experiment we did in mice. We treated for about 4 days with recombinant protein, stopped treating with the recombinant protein, and then look at the return of follicles. So primary follicles they return to their normal levels within 5 days, secondary follicles within 10 to 15 days, and then finally they reach the antral stage around 30 days after you remove the AMH. And so we can basically think about this like a sort of a dam that opens, right? So you have a rush of water, and you just have to time it right. So if you trigger at day 15 when they just reach the secondary stage, you get no oocyte because they're just not ready to be stimulated. You wait 30 days when the follicles reach the antral stage. Now all of a sudden you get a very large cohort of recruitable follicles, and you get about 3 times as many eggs as you would normally in a control. So this way we can actually collect and get greater ovulation yield with our control ovarian stimulation. And we've done this also in the context of diminished ovarian reserve in mice that were pre-treated with strong essentially chemotherapy to deplete their ovarian reserve. We can still get upwards of 2-fold more oocyte that way. And the clinical protocols that we're hoping to test shortly is basically similar to what I showed you in mice. Is you treat with AMH for about 30 days before, you wait for the follicles to resume their growth, and then you undergo standard control ovarian stimulation protocol, and then hopefully you get more eggs on the other side of it. And you may not need this for the regular IVF patient or ART patient, but in terms of women, say that have diminished ovarian reserve, even getting 1 or 2 more eggs may be clinically meaningful. And then lastly, I'll give you a little bit of an overview of how we think that this may be actually useful also for ovarian aging and menopause. And this is studies that we've just recently, well, that's not really true. I've been working on it for a while, but I've just recently started to really come together, and in part because we've learned so much from the cat. So some of those first studies that we did were over 10 years ago in cats. We've got cats that have been treated very long term. We're just now beginning to look at their ovaries, and what we find is very interesting in terms of not only preservation of the ovarian reserve, but also in terms of their continued hormonal production of estradiol. So you all know this, ovaries are a strange organ in that they age faster than most other organ. And if your heart or your lungs stop working at age 51, a median, that'd be a very bad thing. So it's very strange how fast ovaries age, and once you lose their hormonal contribution, it's associated with a lot of different disorders and problems with glucose, cardiovascular health, neurocognitive health, immune function, sexual dysfunction, bone density. So there's, as we live longer, there's more time spent in postmenopausal period, and many of these side effects of menopause, are clinically a burden and something that we want to address. And HRT is part of the solution, but the ovary produce a lot of other hormones on top of estradiol and steroids. There are many, many other things that the ovary provides that may be implicated in some of these deficits. So it's been long known that AMH and menopause and ovarian aging are correlated. And if you look at the number of primordial follicles over time, I'm sure you've all seen this. You have a kind of a linear reduction of ovarian reserve as you age, and then an acceleration as you approach the age of menopause. And eventually, you run out of primordial follicles around the time of menopause, and then they're nearly completely depleted at this time. And AMH has a similar relationship, although it's highly variable between women during their reproductive years. It also goes down with time and essentially reaches near zero concentration around the timing of menopause. So it's at the very least AMH and ovarian aging are correlated, and ovarian reserve are correlated. But then there's some interesting association. So on the right, you see basically the different percentiles of women in terms of AMH that they have in circulation. And if you look at their AMH level, it's predictive of the onset of age of menopause. So the higher the AMH, the later the distribution of age of onset of menopause, and vice versa. If you have low AMH in your reproductive years, you have early onset of menopause. And it's not just an association, because what we find is that in PCOS patients where there's elevated AMH, menopause seems to occur 2 to 4 years later. So there's an interesting association here. Maybe AMH is actually having superficial or high levels of AMH may actually be part of the mechanism of the delay. And so this is how we kind of were thinking about it, at least initially, is that you've got depletion of your ovarian reserve, you run out of ovarian reserve, you run out of growing follicles, and then you run out of steroids, and that's basically what menopause is. And then maybe with higher levels of AMH, either through treatment or I guess through conditions such as PCOS, you can restrict the size of you can reduce the rate of activation of primordial follicles, which could allow you theoretically to have more ovarian reserve left later in life. And maybe that would be enough to have a slight delay in the onset of menopause, maybe something in the order of 2 to 4 years, as you see with PCOS. But it wouldn't affect your growing follicle pool, because the size of the ovarian reserve is not limiting to the size of the growing follicle pool. It's vastly larger until basically you approach the age of menopause. So with a slightly increased AMH level, you probably wouldn't notice it hormonally, and then maybe that would translate in a small delay in menopause. And then that's the other question that we have. Is also, what about follicle pausing? Like maybe you can, if you can stop the development of follicles and freeze it in time, maybe you can stick around longer and provide hormonal support. So those are two different mechanisms by which AMH may ameliorate the side effects of menopause. And so we've begun studies to test these hypotheses. So it's very difficult to do menopause studies because most animals don't undergo menopause, but monkeys and whales do. And although I don't have access to whales, I do have access to monkeys through my collaboration with Adam Krieg at OHSU. And so we've developed a gene therapy vector. We've cloned the rhesus monkey AMH. We've modified it to increase activation. We've purified the protein. It's essentially identical to the human protein in terms of its cleavage and its activity. And we just dosed our first 3 monkeys, and we're hoping to follow them over the years and see how how their reproductive system ages and what's the effect on reproductive hormones, etc. So in summary, I showed you that AMH can achieve durable contraception in cats. We have clinical trials that are pending. We're well on our way to the small molecules that can replicate this action. And the difficulty here is getting them across that hurdle into the clinic in terms of drug development. It's a very hard thing to do academically. And then finally, if we do have treatments where it be recombinant protein or small molecules, there's, I think, many opportunities for clinical applications. I haven't even talked about oncofertility, but I showed you a little bit about ovarian stimulation, DOR, reproductive longevity potentially, even maybe treatments for PCOS. So with that, I'd like to thank you all for coming here, and I'd like to thank my team for doing this work, particularly Marie Meinson, who does the small molecule contraceptive design, and Philippe Godin, who's doing the gene therapy treatments, and many collaborators. I highlight Tom Thompson, but Bill Swanson and Lizzie Van Sant at Cincinnati Zoo that are helping with the cat studies. So with that, I'm happy to take questions.